floor.
I will get rid of that really quickly just in case that is an issue later.
Yeah, just slide it aside or something.
There we go. Okay. Thank you, Dr. Greer, for the introduction, and thank you everybody for coming and joining on Zoom. So I'm just here to go over the progress of my project, the identification and characterization of fat kinase as a novel regulator of PD-L1 stability and activity in cancer. So before I go into my presentation, I just want to go over a little overview on what I'm going to be talking about today. So first, just some background information on PD-L1 structure and function, PD-L1 in immune evasion and immunotherapy, and some post-translational modifications. After that, I'll go into my hypothesis and specific aims, my results, some of my future directions, and then just a short summary of my project and its significance. So just to start us off, I'll be going over some background information. So programmed death ligand 1, also known as PD-L1, was first discovered in the 1990s to be a part of the B7 family. So the B7 family consists of structurally related cell surface protein ligands, which bind to the CD28 family of receptors. One example being programmed death receptor 1 or PD1. So, and these are typically found on lymphocytes and regulate immune responses via co-stimulatory or co-inhibitory signals. So PD-L1 can be found on a variety of cells, some hematopoietic being dendritic cells, T regulatory cells, or macrophages, and some non-hematopoietic such as epithelial cells. So we see a little figure of PD-L1 right here, but what is its exact structure? So PD-L1 is a type 1 transmembrane protein that binds to its receptor PD1 on its extracellular domain here. And along with the extracellular domain, there are two other domains, one being the transmembrane domain found here that helps anchor PD-L1 to the cell surface, and then the intracellular domain that binds to cellular proteins within the cell. So now that we know its structure, what exactly is PD-L1's function? So as I mentioned before, PD-L1 binds to its receptor PD1. But let's start before that. In a normal immune system, after T cells are activated in the lymph nodes and traffic to a point of infection or injury and interact with infected cells through the T cell receptor or the MHC complex and kills the infected cells, they will continue to proliferate and activate just until the infection is gone. So once the infection is completely gone, T cell activity needs to stop. And one way that it's done is through the PD-L1-PD1 interaction. So as we see here, these cells, the T regulatory, macrophage, and dendritic cells, as I mentioned as examples before, express PD-L1 on their cell surface to interact with PD1 on T cell surface. So this interaction deactivates T cell activity and will eventually lead to apoptosis as seen here. But what would happen if PD-L1 wasn't there? There we go. So once again, after cytotoxic T cells kill an infected cell and continue to activate and proliferate, but they wouldn't have anything to stop it. So there would be no PD1, PD-L1 interaction, which would continue with T cell proliferation and activation. And this activation will lead into an increase in immune responses, autoimmunity, and inflammation. So therefore, the PD1 and PD-L1 axis is essential for central and peripheral immune tolerance. And due to its function, this axis has been coined as an immune checkpoint. But how does PD-L1 and its function relate to cancer? So preclinical studies have shown that PD1 and the PD-L1 axis could be a potential mechanism in which cancer uses to escape T-cell anti-tumor responses, as PD-L1 has been found to be expressed on a vast amount of cancer cells, particularly in solid tumors such as lung cancer, colorectal cancer, and breast cancer. So as seen here, the T cell receptor found on the T cell surface binds to the MHC complex that's found on a tumor cell due to the recognition of a neoantigen presented by the cancer cell. So this then activates cancer cell killing from the T cell. But cancer cells have adapted ways to express PD-L1 on their cell surface to bind to PD1 on T cells, which would then induce T cell death and eventually immune evasion. So due to this function, the PD1 and PD-L1 axis has become the target for cancer therapy research. So in 2018, James P. Allison and Tasuku Honjo received a Nobel Prize for their work that led to the discovery of immune checkpoint inhibitors, also known as ICIs. And in 2014, the very first anti-PD-1 therapy, pembrolizumab, was FDA approved. So the goal of this immunotherapy was to boost anti-tumor responses and stopping immune evasion by blocking the PD-1 and PD-L1 interaction. So as we see here, before a treatment is given, the PD-1 and PD-L1 interaction is able to occur, which would eventually inhibit T-cell killing. But if the anti-PD-1 or anti-PD-L1 therapy were to be administered, this interaction would no longer be able to happen, as either the ligand would be blocked by anti-PD-L1 or the receptor would be blocked by anti-PD-1. So as of right now, immune checkpoint inhibitors have been approved for more than 25 types of cancer, one being melanoma, which has seen some of the best response rates to anti-PD-L1 therapies, but there are some limitations to this. Only very few cancers have actually exceeded a response rate of 40%, and only few patients have reached a complete response. So as we can see here, for example, for colon and rectum cancer in men and women, there's more than a 50% response rate to anti-PD-1 and PD-L1 therapies. But despite this success, there are cancers such as prostate in men and breast in women that have only reached a 10 to 20% of overall response rate. So many tumors have been found to have intrinsic or acquired resistance to anti-PD-1 or PD-L1 therapies, and these molecular mechanisms are still largely unknown. Thus, there's a need to boost efficacy of anti-PD-L1 therapies, and recent studies have shown that post-translational modifications play an important role in anti-PD-L1 therapy responses. So post-translational modifications such as phosphorylation, glycosylation, and ubiquitination are integral for protein regulation. Recent studies have shown that post-translational modifications are essential in PD-L1 expression, but also the PD-1, PD-L1 mediated immune evasion. So given the role that kinases have in the regulation of proteins, investigations began to see if there were any protein kinases that directly modify PD-L1. So as of right now, there have been a few identified, such as GSK3 beta, AMPK, CK2, and JAK1 that have all been found to phosphorylate and regulate PD-L1. So I just wanted to show a quick example on how PD-L1 expression is affected by post-translational modifications. So as we see here, when IL-6 meets its receptor, it activates JAK1. So this activation of JAK1 leads to the association with unglycosylated PD-L1, and this association allows for JAK1 to phosphorylate PD-L1 at the tyrosine 112 site, and this phosphorylation promotes the association with STT3A, leading to glycosylation. And the glycosylation of PD-L1 allows for the protein to be stabilized, which would then lead to the membrane expression of PD-L1 on the tumor cell and eventually interacting with T-cell PD-1, leading to immunosuppression. But when IL-6 is inhibited and its receptor is not activated, there is no JAK1 activation, meaning that it no longer associates with unglycosylated PD-L1, therefore no phosphorylation, no STT3A association, and no glycosylation. And without glycosylation, the protein is no longer stabilized, leading to degradation, subsequently meaning that there is no PD-L1 to be expressed on the cell surface of the cancer cell, leading to an enhanced T-cell killing effect. So to date, there are about 560 kinases that have been identified in the entire human kinome, but there has been no systematic screening that's been done to identify kinases that regulate PD-L1. So this has made me ask the following questions. What is the mechanism behind PD-L1 upregulation in cancer? What other kinases could have a role in PD-L1 stability and activity? And then finally, how can we identify other kinases that regulate PD-L1? So now this leads me to my hypothesis. So now I'll just be discussing my hypothesis and specific aims. So my hypothesis is that given the function of protein kinases, we hypothesize that novel kinases identified by systematic screening may regulate cancer cell immune evasion through regulation of PD-L1 stability. And with this hypothesis, I have three aims I have been investigating. First is to identify and validate kinases that regulate PD-L1 stability by a kinome-wide protein kinase inhibitor screen. Next is to characterize FAC as a novel regulator of PD-L1 stability in cancer cells. And then finally, to understand the interaction of FAC and PD-L1 in immune evasion. And so now I will just go on to my results with these three aims. So starting with my first one, to identify and validate kinases that regulate PD-L1 stability, I have three sub-aims. First is to establish a HEC293A cell line that stably expresses PD-L1 nanoluck. Next is to conduct a high throughput screening for protein kinase inhibitors that may regulate PD-L1 stability. And then finally, validating any of these primary hits through a Western blot analysis. So just to start on to my first sub-aim, to establish a HEC293A cell line. One of the postdocs in our lab, Dr. Prem Kanal, actually started this PD-L1 project. So he cloned the open reading frame of PD-L1 into a PNLF vector to make a construct expression of PD-L1 nanoluck luciferase fusion protein. So the nanoluck tag was put onto the C-terminal end of the protein, as we see here, to measure protein stability through luciferase activity. So it's thought that if luciferase activity went down, protein stability would also go down. So we then transfected the plasmid into HEC293A cells and selected the cells that were expressing the plasmid with hygromycin. So to verify that the cells were stably expressing PD-L1 nanoluck, we did a luciferase assay and a Western blot to validate expression. So as seen here, different clones 1, 2, and 3 successfully express PD-L1 nanoluck at different degrees. So the y-axis here just represents the relative luminescence units, and the x-axis is just our sample, so our control here, and then our 3 clones that we chose for this assay. So as we can see here, luciferase activity of these clones are different, as I mentioned before. So this should correspond with our Western blot that we did to follow up these results. And that is exactly what we saw. So when we look at the luciferase activity, from clone 3 to clone 1, luciferase activity decreases. And that's exactly what we see here in this Western blot. Clone 3 to clone 1, expression of PD-L1 nanoluck also decreases. So now that we have a cell line that was stably expressing PD-L1 nanoluck, we could then move on to the next sub-aim, which was to conduct a high throughput screening for protein kinase inhibitors that regulate PD-L1 stability. So since, as I mentioned, there was no systematic screening of protein kinases that regulate PD-L1 before, that's where we decided to begin. So on day 1, the previously established HEC293A PD-L1 nanoluck cells were seeded into 2 384 well plates. The next day, the cells were then treated with kinase inhibitors and DMSO as a control. And then after 24 hours of treatment to measure the direct effects of the inhibitors, the substrate was added and luciferase activity was measured. And then when discerning a significant suppression, the relative activity compared to the control was calculated, and anything with a 2-fold decrease or more was considered a hit. And then just to quickly acknowledge that the Mount Sinai Hospital in Toronto carried out this screening for us. So after this screening, we were left with a lot of data to look at. So here is the mean luciferase activity between the 2 plates. So each sample had 2 replicates. So to get the relative activity, the mean control of the samples were divided by the mean, luciferase activity of the samples were divided by the mean luciferase activity of the control. And so here is the relative activity of all the samples compared to the control. So I know that looking at a bunch of numbers might be hard to look at, so I do have something a little bit more digestible. So here is a scatter plot of all the samples' relative activity. So anything under this red line here represents a 2-fold decrease, which was considered a hit as it's the most dramatic effect. So from here, 19 hits were identified, and from these 19, 16 of these hits were novel, and 3 of them were already known, which shows that our screen worked. So we decided to focus on protein kinases that are highly regulated in cancer, which cut down our list from 16 to 10. So here was the complete list of all the protein kinases that I was continuing to investigate with. And these 3 kinases here, CK2, ROCK, and GSK3 beta, were the kinases that have been recently published, therefore indicating our screen worked again. So to validate these hits, I then conducted another luciferase assay. So I treated the HEC293A PD-L1 nanoluck cells with 5 micromolars of each inhibitor for 24 hours. Then after the treatment, I measured the luciferase activity. And here were the results of that assay. So on the y-axis here is just the relative luminescence units again, and the x-axis is all of the samples that I used with the kinase inhibitors I was targeting along with the drugs. So for example, GSK3 beta was the inhibitor that I was was the protein kinase that I was trying to inhibit, and CHIR 99021 was the inhibitor being used. So as we can see, only 6 of the 10 inhibitors tested had a significant decrease compared to the control, along with the other 4 kinases that were tested. And then from these 6, the bars in orange now are the three kinases that were previously verified, once again showing our assay worked. And so the other 3, FAC, CDK149, and ALK5 are all new. So these 6 will be the focus of my next sub-aim, which was to validate the primary hits of from the luciferase assay with a Western blot analysis. So here are the results for the Western blot after I treated the HEC293A PD-L1 nanolux cells with 5 micromolars of each treatment for 24 hours and using the Ponzu S staining as a control. So after treatment, we can see that FAC, ALK5, and CDK149 had very low PD-L1 expression compared to the DMSO control, but also the 3 positive controls that we were using earlier. So out of these 3 kinases, my main focus right now is FAC, as it has been found to be involved in many cancers. But before moving on, I wanted to validate that these results were true. So I then conducted a dose-dependent analysis with one of the positive controls. So I did a follow-up dose-dependent luciferase assay and Western blot with the GSK3 beta inhibitor that I used in the initial luciferase assay to validate that my previous Western blot results were true. So as we can see here, as the dose of the GSK3 beta inhibitor increased, the luciferase activity decreased up here, and that corresponded with my Western blot results, showing the control compared to 2.5 micromolars of the GSK3 beta inhibitor. So validating that my Western blot results previously were true, FAC was then a reliable target to continue the rest of my experiments with. So now the real question is, why is FAC our target? So focal adhesion kinase, also known as FAC, is an intracellular non-receptor tyrosine kinase. It has previously been shown to mediate interactions with many proteins and to be associated with many cellular functions in normal and cancerous cells. Some of these functions include cell motility and adhesion, survival, lymphovascular invasion, epithelial mesenchymal transition, and cell migration. And FAC has also been found to be highly regulated in cancers, such as breast, lung, and head and neck cancer, just to name a few, therefore becoming our target. Now that aim 1 is wrapped up and we have a kinase to focus on, we can then move on to the second aim, which is to characterize FAC as a novel regulator of PD-L1 stability in cancer cells. And this aim also had 3 sub-aims. The first one was to validate FAC as a novel regulator of PD-L1 stability, see if there was an interaction between FAC and PD-L1 in vivo, and then see if FAC regulates membrane PD-L1 stability. So first, going on to the first sub-aim, to validate FAC as a novel regulator of PD-L1, after finding that the FAC inhibitor I previously used decreased PD-L1 expression, I did another dose-dependent luciferase assay and a Western blot in my HEC293A cells and treated those cells for 24 hours with varying doses. So as I mentioned, I administered different doses of the FAC inhibitor PF431396, which I used for my initial luciferase assay and treated the cells for 24 hours. So as we can see here, as dose of PF431396 increased, luciferase activity of PD-L1 nanologue decreased along with the Western blot here. So this shows that the FAC inhibitor downregulates PD-L1 stability. But since I used this inhibitor before, I wanted to make sure that these results were consistent with other FAC inhibitors. So to verify this, I did the same experiment with a different FAC inhibitor, defactinib. So here are my results for defactinib. Once again, I treated the cells with varying doses of the inhibitor for 24 hours. And as we can see, as the dose of defactinib increased, once again, luciferase activity decreased, along with our Western blot results. And just to quickly note that the 10 micromolar treatment of defactinib had the most significant PD-L1 decrease. But overall, this shows that defactinib, the FAK inhibitor, downregulates PD-L1 stability. And this shows that FAK inhibitors had consistent results to one another. But since I was using kinase inhibitors and that had a potential for having off-target effects, which could further influence PD-L1 instability, we did a knockout of FAK in 2 triple-negative breast cancer cell lines just to make sure that it was FAK itself regulating PD-L1 and none of those off-target effects. So we used the cell lines MDA MB231 and BT549. So to do this, the CRISPR system was used, and I wanted to go over that protocol really quickly. So to start, the cancer cells MDA MB231 and BT549 were seeded in a 6-well plate. Afterwards, 1 well of each cell line was treated with a virus that expressed 3 different clones of FAK guide RNA to knock out FAK function. And then after a few days, the proteins within the cells were extracted using the RIPA buffer. As it's been shown, the RIPA buffer has better efficiency in extracting membrane bound proteins. And then finally, the samples were visualized using a Western blot analysis. So here were the results to that. So as seen in both cell lines, when FAK was present and there was no guide RNA, the PD-L1 expression was more defined. But when FAK was knocked out in both MDA MB231 and BT549 cells, PD-L1 expression decreased. So we see here that the knockout of FAK causes a decrease in PD-L1 stability. I then wanted to see what would happen on a gain of function. So if overexpressing FAK causes an increase in PD-L1 expression. So that's exactly what I did next. So this time I used the dox-inducible system. I treated lung cancer cells with H1299 cells that were previously established by PREM with dox for up to 3 days. So just to see if the overexpression of FAK would cause an overexpression of PD-L1. And as we can see from the results, as FAK expression increased, so did PD-L1 expression. Therefore, FAK overexpression increases PD-L1 stability. And the main reason why I wanted to use lung cancer cells in this experiment was because, as I mentioned before, FAK has been found to see regulated in different cancer types, breast and lung cancer being a couple of them. So I wanted to see if the relationship between PD-L1 and FAK was similar across other cell types and rather than just one. So now that both the loss of function and gain of function experiments showed that FAK regulates PD-L1 stability, I can then move on to my next sub-aim, which was to see if there was interaction of FAK with PD-L1 in vivo. And to do this, we did a co-immunoprecipitation assay. So I really quickly wanted to go through my co-IP protocol. So this analysis is to essentially look at protein interactions using a Western blot analysis. So to start off, my proteins of interest, FAK or PD-L1, were transfected alone or together in HEC293A cells. And so afterwards, I extracted the cell lysates, and after extraction, I added either an anti-FLAG or anti-MYC antibody along with magnetic beads into the tube to be able to isolate the complex and pull down the proteins of interest. Afterwards, I washed the cell lysates with the magnets just to make sure that the proteins of interest were the only ones left in my solution. And then next, the proteins were eluted so that they were no longer attached to the beads. And then we used a Western blot analysis to see if there was a protein complex between our proteins of interest, FAK and PD-L1. So now we have samples where PD-L1 and FAK are present alone or together, as we see up here, but also in the cell lysates as a control. So in this plot, we were pulling down PD-L1. And in the first column here, we expected to see little to no interaction just because FAK was not present, but PD-L1 was, and we saw exactly what we expected. In the second column, with PD-L1 absent and FAK present, we can see that there was minimal interaction with the FAK that was present and the PD-L1 being pulled down. And then finally, when both plasmids were present, we see an interaction between FAK and PD-L1. So this shows that PD-L1 does interact with FAK. But just to verify these results, we wanted to do the exact same thing, but in reverse. So this time, we were pulling down FAK, and we saw pretty much the same results. When PD-L1 was present, we saw minimal interaction with FAK being pulled down. When FAK was present, but PD-L1 wasn't, there was nothing. But when both were present, we see an interaction. So this means that FAC and PD-L1 interact with each other, but there was a little bit of a problem with this. So since the results were from cells that were transfected with PD-L1 and FAC, we wanted to verify if this was true endogenously. So the next steps was to observe endogenous expression of FAC and PD-L1 in MDA MB231 cells. So here we have 4 milligrams of the MDA MB231 cell lysates that, and here we were pulling down FAC. And so after blotting against PD-L1 and FAC, we can see that there is a strong interaction between the two. So once again, like the previous co-IP, we want to do the same thing, but in reverse, just to verify our results. And we saw the exact same thing after pulling down PD-L1, that there is an interaction. But here it looks like that the interaction of pulling down PD-L1 was not as strong after pulling down FAC. And so the reason for this is because FAC may interact with other proteins before the antibody could actually attach. So therefore, the FAC wouldn't be able to interact with that antibody. And since in this co-IP, we were just trying to detect the PD-L1 FAC complex, nothing would be detected, which could be the reason for this lower signal. So since we were able to see that FAC does interact with PD-L1, we could then move on to my next sub-aim. And this sub-aim is just to see how FAC regulates membrane PD-L1 stability. And this was done using a fluorescence-activated cell sorting analysis, or a FAST analysis. So now I just wanted to go through the protocol of my FAST analysis. So the purpose of this experiment was to observe how membrane PD-L1 expression changed after going under a 24-hour treatment. And I used FAC inhibitors for this. So I used the HEC293A PD-L1 nanolux cells and treated them with either DMSO as the control, 5 micromolars of PF431396, which was the FAC inhibitor used for my initial luciferase assay, and 10 micromolars of defactinib, as if we remember from AIM 2.1, 10 micromolars of defactinib had the most significant decrease in PD-L1 expression. So after 24 hours of treatment, I counted 1 × 10⁶ cells for each sample and resuspended the cells in a cell staining buffer. I then added a fluorescently labeled anti-human PD-L1 antibody at a 1:100 dilution and let that incubate for 30 minutes. Afterwards, I washed the cells and resuspended them in a cell staining buffer again and strained them in a 35 micrometer strainer, just so we made sure we had single cells and not a big clump of them. And then finally, the samples were analyzed with the Cytoflex machine to measure membrane PD-L1 expression on the cells. And then IgG was also used as a control to show that the fluorescence showed that the fluorescence that was being detected for PD-L1 was true. So here are my results for my HEC293A PD-L1 nanolux cells. So on the y-axis here, it is supposed to represent the count, but since the count for all the cells were the same with 30,000 cells, I just omitted that, but labeled them just so it's easier to follow for the condition that was used. And then on the x-axis here, it's the membrane PD-L1 that is being detected on the cell membrane. So each of these curves represent how much membrane PD-L1 expression is present on the cells in each sample. So if PD-L1 were to increase, there should be a shift to the right towards 10⁷. But if it were to decrease, there should be a shift to the left to 0. So as we can see from the image alone, there was a slight shift after 24 hours with the 5 micromolars of PF431396, but a much larger shift after 10 micromolars of defactinib for 24 hours, which from this image shows that PD-L1 membrane-bound PD-L1 does decrease after FAC inhibitor treatment. But to further analyze the data, I calculated the relative change of the median, and I chose the median just as it's the most middle point of all the data that was given. So I set DMSO as 100% because since there's no treatment, every single cell in that sample should be expressing membrane-bound PD-L1. And to calculate the relative change, I divided the medians from the other conditions by the median of DMSO. And so as we can see here, there was almost a 40% decrease of cells that were present that were expressing membrane-bound PD-L1. So this is consistent with the previous Western blot that we saw in 2.1 for defactinib, as PD-L1 expression decreased for after 10 micromolars of treatment. So we can see that FAC expression regulates membrane PD-L1 stability. So from this aim, we learned that FAC is a novel regulator of PD-L1 stability, FAC and PD-L1 interact with each other, and that FAC regulates membrane PD-L1 stability. So this allows us to move on to the final aim, which was to understand the interaction of FAC and PD-L1 in immune evasion. So if we remember from what I mentioned at the very beginning of my presentation, is that cancer cells are able to use PD-L1 and express it on their cell surface to avoid anti-tumor responses. So we wanted to see if FAC plays a role in that due to it affecting PD-L1's membrane stability. So I did this by conducting a T-cell killing assay. So I wanted to just discuss the protocol for my T-cell killing assay first. And the purpose of this assay was to track T-cell mediated cancer cell death in the presence or in the absence of FAC. So I used wild-type MDA-MB231 cells and the previously established FAC knockout, and I put them in a 96-well plate in 3 rows. So 2 rows having the wild-type MDA-MB231s, and the last row having the FAC knockout. I then added the previously activated PD-1 expressing JERKIT T cells to rows 2 and 3, as the first row of the plate was just to follow cell growth without T cells. Afterwards, I then added an apoptosis reagent, incucyte caspase 3,7 green dye, to each well as the dye is supposed to cross through the cell membrane and is cleared by activated caspase 3,7. So when the cell dies, it releases a fluorescent green dye, which allows us to actually count the cells that are dead in each image that's taken by the machine. And then finally, after a 30-minute incubation time, I monitored T-cell killing using the CellSite X imaging system for 24 hours. So before going into my results for my T-cell killing assay, I want to touch on the activation of T cells first. So as I mentioned, I added activated T cells that were expressing PD-1 to my plate. And I had to activate the T cells with forable myristal acetate, PMA, or phytohemagglutinin, PHA, for 48 hours. So after a 48-hour treatment, I collected the cell lysates so we could observe PD-1 expression. And as we can see, using beta-actin as a control, without the PMA PHA treatment, there was no PD-1 expression. But after 48 hours of the treatment, PD-1 expression was quite strong in the JERKIT T cells. So now that my JERKIT T cells are activated and expressing PD-1, we could then track T cell killing. So here are the results after 24 hours. I did only show the first 12 hours as they are the most significant. So the x-axis here were just the time points of each hour. And so I took 1 image every 30 minutes. So it was 2 images per hour. And then the y-axis shows the fluorescent cell count. But if we remember from when I was explaining my protocol, it's actually dead cells because the dead cells fluoresce due to the apoptosis reagent that I used. So just to go over the results, so the black line here is the control, and this is the cell type that only had the wild type MDA-MB231 cells without the JERKIT cells. So these were just to track cell growth and shows very minimal cell death, which is what we expected. Next is the blue line, which is the MDA-MB231 cells with the JERKIT T cells. And these cells are for us to keep track of normal cancer cell and T cell interactions prior to treatment. And then finally, this orange line here is the MDA-MB231 cells with the FAC knockout. And these cells are to help us keep track of T cell killing with the absence of FAC. So subsequently, a reduced membrane PD-L1. So as we can see here, the knockout or the orange line had more T cell killing than compared to the wild type, which can help us lead up to the conclusion that a FAC knockout leads to increased T cell killing. The thing is, these are just preliminary results right now, as I'm still trying to optimize my T cell killing protocol, but these results as is look very promising for my future experiments. So now that I have wrapped up what I've done so far with my 3 aims, I can go into some of my future directions. First, so along with the triple-negative breast cancer cell knockout in 2.1, I wanted to conduct an siRNA knockout in my HEC293A PD-L1 nanolux cells. And then I also wanted to do a big screening of more triple-negative breast cancer cell lines for FAC expression, subsequently how the relationship between FAC and PD-L1 in those cells. Next, for 2.3 was to observe how membrane stability, PD-L1 stability changes after the HEC293 PD-L1 nanolux cells have an siRNA FAC knockout. And then finally, just further optimization of my T cell killing assay and eventually a rerun of those experiments as well. So now I can just go on to my summary of my findings and its significance. So just to see what I've learned so far throughout my projects, we have successfully conducted a systematic screening of all protein kinases that could be involved in the regulation of PD-L1 stability and activity. We identified that there are 16 novel protein kinase regulators that played a part in PD-L1 suppression, and 10 of those were highly regulated in cancer. We identified a relationship between FAC expression and PD-L1 expression, identified that FAC regulates PD-L1 stability and membrane PD-L1 stability, identified that there's an interaction between FAC and PD-L1, and then identified that FAC plays a role in T cell immune evasion. But what does this all mean? So these findings give rise to potential targeted treatments for cancers that do not have high response rates with anti-PD-L1 therapies, for example, triple negative breast cancer. But this also could result in more efficacious anti-PD-L1 therapies in combination with FAC inhibitors. And just finally, I would like to thank Dr. Yang for his support and guidance throughout the past year and a half. Thank you to the members on my supervisory committee, Dr. Craig and Dr. Goudreau, and the program coordinator, Dr. Greer, for their advice and guidance throughout my master's. I would also like to thank Mount Sinai Hospital for conducting the screening of the kinome. And finally, I would like to thank the members of the Yang Lab for their daily advice and support. Thank you for listening. Yeah.
Okay. Questions, please.
That was an exciting talk, a great project. Struck me that one of your hits was FGFR, and there are certain cancers like one I work on called bladder cancer that has a subset of patients who have activating mutations in FGFR3 in particular. Does this mean that maybe they have what do you think the FGFR targeted inhibitors would do versus PD-L1 therapy versus both in those patients?
That's a very good question, just because I know that here from, let me scroll over. I believe it was this figure here. Oh, no, it's not. Oh, yes. So here for urinary bladder cancer, because there's only about a 20 to 30%, it looks like, in men at least, for PD-L1 response. So, from my results, at least, it didn't show that FGFR was a huge a decreased PD-L1 by that much, but there is worth looking into seeing if putting anti-FGFR and anti-PD-L1 therapies together might work, just because there have been, for example, there was a paper published that used PARP inhibitors to increase PD-L1 expression and then use anti-PD-L1 as in conjunction, and saw that there was a decrease in tumor growth. So maybe potentially, even though FGFR didn't have a huge impact on PD-L1 expression, as seen here, at least a decrease, maybe adding anti-FGFR, anti-PD-L1 therapies together might show some positive results for treatments.
Can you go to the figure where you did your scatter graph on your results for screen? Yeah. So you were focused on things down below the line. Mm-hmm. What about this spot?
Yeah. So these were actually the list of kinases that we found to increase PD-L1 expression. Right now, that's not in the scope of my project, but it's definitely, we have thought about that and thought that it is worth looking into. Like, as I mentioned before, it has shown that increasing PD-L1 expression and then adding anti-PD-L1 therapies has increased the efficacy of anti-PD-L1 therapies. So definitely looking into these is a good next step as well.
Sure. So was that EGFR, that one that was way up there?
I'm not too sure exactly which one.
Well, never mind. This is actually where I was going with this. So this screen, you use a bunch of inhibitors.
Mhm.
So what do each of those inhibitors, do you just think it's one kinase that they inhibit, or?
There's, sorry, I have another list. Right here. So these inhibitors show that so mostly at least it's at least one, but it does show that there is an increase. I'll use here, or there is some with multiple targets. For example, CDK149, that's CDK1, CDK4, and CDK9. So we don't exactly know which CDK it is, or if it's in conjunction with two of those or three of those. So it definitely could be more than one thing being hit at once. So when we were trying to find other inhibitors to use, we tried to look at other targets that they had just to like see how dirty the inhibitor was or how clean it was. So we tried to get, we did try to get as much like specific as we can, but there's definitely inhibitors that we use that could have other effects or other targets that could also influence PDL1.
Okay, that's a good answer, because I think probably the people that put together this panel gave a lot of thought to that.
Mhm.
It's use the term dirty. There are a lot of kinases that are quite cross-reactive.
Yes, yeah. Yeah, that was a big issue. For example, for the CDK149, another like a next step that we've been talking about is looking at that specifically. So we have a list. So I was looking for a bunch of drugs that inhibit CDK149, but I could only come up with this because a lot of CDK inhibitors also inhibit a lot of other stuff as well. So I tried my best to find as many that only inhibited CDK1, CDK4, or CDK9, or them in conjunction.
We may have a couple of those.
Oh. That's good to know.
I got lots of questions, so somebody.
Yeah, so congratulations. I mean, one of the things that strikes me is you've done a lot, and you've done a lot of different methods. So that's fantastic. You should be very proud of that. I mean, I had questions about many of them because it's sort of details that you didn't have time to talk about. But your cell killing experiments towards the end, tell me a little bit more about that. So you throw in Jurkat cells, which are CD8 positive T cells, I suppose. So how do you know that they're going to kill your tumor cells? I mean, there has to be a TCR interaction with an antigen. So is there history that says that Jurkat cells will kill B cells?
That's a really good point to be brought up. So with the Jurkat cells, so they are like just very broadly used over many other screenings to do with T cells. So that was one reason why we chose it. But another reason why was back in 2018, a paper that was actually published in our lab and our cancer research used a similar assay. It wasn't exactly the same, but they used Jurkat cells to look at Jurkat T cell activity and T cell killing with the luciferase assay, and it shows that Jurkat cells, after being induced by like PHA and PMA, had an increase in killing versus cells that didn't. I hope that.
So what happens to the Jurkat cells when they're induced to kill? How do they change their molecular surface profile? Is that what happens? There's something. So usually the killing would be mediated through a distinct interaction with TCR.
Yes,
and
yeah.
And the peptide is presented by MHC. But maybe is there something else happening here?
From what I know for the Jurkat cells, it should be the TCR and the MHC complex as well. One issue that we've actually been having with the T cell killing assay, which is why we need further optimization, was the activation of the Jurkat cells. So for example, seen here, this looks pretty promising, but the y-axis showing cell death isn't huge. Like it's very low numbers. So after talking to Dr. Yang and looking into it more, we saw that there was, sorry, to answer your question, I believe it is the TCR and MHC complex that does occur, but the activation of the Jurkat cells is also really integral to just having good results for these. So there was a paper back in 2016 that was published that had a really similar experiment. So I always talked about activating the T cells. And like I said, I use PMA and PHA, but that showed that it doesn't really mimic natural processes that we want for the Jurkat cells and doesn't also have a sustained response. So to get through this, looking at this paper, they use anti-CD3 and anti-IL-2. So anti-CD3 activates the T cells by mimicking natural TCR engagement, kind of like what you were saying with the T cell receptor. And then IL-2 helps promote the survival and proliferation of it, which PMA and PHA just didn't do, unfortunately. So yeah, it is supposed to go through like that natural pathway that it usually does, but we have to just artificially activate it with these cells.
Thank you.
Go back to your graph with the killing assay.
Yeah.
So what happens to FAC knockout cells? What do their morphologies look like compared to?
I remember your question from last year because this was the you asked us last year. Because after you asked that, after you asked that, I made sure to look at it, and honestly, the morphology is quite similar to the wild type cells, and especially since I had to look at them side by side for so long, it was like there was no difference at all. Like when I looked at them, the only way I knew that they were different was because of my labels. Like just looking at them, I wouldn't be able to tell that they were different.
Because you know why that's important. They should not be able to remodel their focal evasion. They should stick like glue. Do you notice that they're harder to trypsinize, or that
they are a little, sorry, I say for trypsinization usually. So for cancer cells, typically it's 5 minutes for like the HEC293 cells, but for cancer cells it's a little bit longer. And then for the cells that with a FAC knockout, I find that I have to do a little bit longer and a lot more pipetting just to separate them and get them all off. Yeah.
You'd expect for this. So that gets to this assay. So when you have two different cells, wild type and knockout, that may have different adhesive characteristics. Do you know that increased signal is really because you're seeing more dying cells, or you're just more cells are retaining on the surface of the plate?
Yeah, that's a very good point, and a way that we were actually planning on kind of avoiding that, just because we wanted to make sure that the reason for the cell death was because of T cells, but also like let's say there was T cell death was because of the PD-L1 PD-1 interaction. We wanted to add an anti-PD-1 or PD-L1 antibody, like a fluorescently labeled one, to be able to visualize that interaction in the images as well.
Well, I was thinking more of some other way of instead of just counting fluorescent cells, counting a ratio of those that are fluorescent, those that are not.
Oh yes, actually, that is something that we did want to do as well. So because we were thinking about that, and in the same paper, as I mentioned earlier, by Lai et al. In 2016, Dr. Yang and I talked about purchasing the M-cherry plasmid, which is a derivative of RFP, to infect cells. So I made a lentivirus with HEC293 T cells so that the live cells are fluorescing red, and so I transfected those into some triple negative breast cancer cells as MDA MB231 seen here, and BT549. So instead of just hoping to look at the fluorescent cell count that the machine gives us, we can actually count the cells. And that's exactly what we want to do. We want to do a ratio of the live cells or red cells versus.
Given what FAC knockout should do to cells, it's a good idea to do something like that. Other questions? Go ahead.
So really impressive experiments that and also I really like that you find really methods that I don't do all of that research. The question I had was the slide that you had like the Kinase bit.
The scatter plot one?
Yeah.
Yeah.
So I guess like all the ones that have the receptor, or like most of them are kind of like all together. Like is there like an order that they were like placed in?
For like the x-axis?
Yeah.
I think those were just the inhibitors itself. I don't think there was a real order to where they were put in. I think it was kind of like this, but a little more bunched up because there were just so many. Yeah.
Next. Oh, we still got a few minutes here.
I saw Dr.
Are you able to make any connection between FAC and the glycosylated glycosylation of PD-L1. And you said that that was partly responsible for the stability of PD-L1.
Mhm.
So is there a known or a possible connection between fat kinase activity and glycosylation of proteins?
As of right now, there isn't. From what we think currently, what could like how FAC modifies PDL1 is through phosphorylation. Just because FAC has been seen to phosphorylate, autophosphorylate itself, it phosphorylates it's called NANOG. It's to help with embryonic stem cell renewal and cancer progression. So we think that the activity is more so through phosphorylation, but that's a very good point that you bring up because phosphorylation can actually increase PDL1 stability or decrease depending on it. So like as I showed with JAK1, it increases stability, but for the kinase, for example, AMPK5, it phosphorylates PDL1 to stop glycosylation and to inhibit the glycosylation, leading to the degradation by the endoplasmic reticulum associated. Yeah. So we're not too sure exactly what FAC does to PDL1 yet, but we think it'll be phosphorylation, and that's kind of the way we're going to go into. But just seeing how it stabilizes as well is also another avenue that we have to go to as well.
So you mentioned in your introduction that there's a tyrosine on the cytoplasmic domain 112 or so that is a JAK target.
Mm-hmm.
So do you know where FAC phosphorylates? Or do you know that FAC phosphorylates PDL?
We don't know yet if FAC phosphorylates PDL1. We think that, so I'm just gonna go over here, actually. Here. This is just kind of a map. It doesn't have everything, but it's a map of the post-translational modifications of PDL1. So we think, so a lot of the phosphorylation sites are in the extracellular domain. So we think that's where the, that's where FAC will also modify PDL1 is in the extracellular domain, but before it gets to the cell membrane and the extracellular domain is outside of the cell.
So but that membrane you had on the other one was,
Oh yeah, no, it's 112, tyrosine 112. So yeah, it should be from this map. It should be over here.
But if you go back to your other map, thought you were suggesting it was on the cytoplasmic. Okay, no, you don't really indicate.
Oh yeah, no, sorry. This image was it just shows that it phosphorylates. It doesn't show the exact.
Okay.
Yeah.
All right. That question. I could talk to you more.
Okay.
So unless there are any other questions, then join me in thanking Asia for a very exciting time.